evening, everyone. Welcome to BioAsk, the show where we unravel the wonders of the biological world. I'm your host, Izan, and tonight we've got a groundbreaking topic that's taking the scientific community by storm: CRISPR-Cas9. Joining us tonight are esteemed biologists, Dr. Elisa and Dr. I. Johannes. Welcome to both of you. Now, let me tell you a snippet of what is CRISPR-Cas9. CRISPR-Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of genome by removing, adding, or altering sections of the DNA sequences. Genome editing is a group of technologies that give scientists the ability to change an organism's DNA. Doudna and Carpentier de Nobilis have published their first paper announcing new enzymes for editing the DNA in June 2012. It was called CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeats that consist of two components, which are the Cas9 protein that can cut the DNA, and another one is guide RNA, which can recognize the sequence of DNA to be edited. The existing of CRISPR-Cas9, the other tool for gene editing technology. We can bring the bright future for treating disease by permanently correcting the deleterious base of mutations or disrupting disease that causing genes with great precision and efficiency, as it is simple to use, cheap, and that makes it revolutionary because it could treat, perhaps even cure the genetic diseases. This could really change a lot of things in our society. Now, let's begin. CRISPR-Cas9 has been making headlines. Dr. Alisa, can you give us, our viewers, a brief on the advantages and limitation of CRISPR-Cas9 on Staphylococcus aureus? Oh yeah, doctor. Please explain about the procedure of it too.
Chen et al. In 2017 states that the Staphylococcus aureus is one of the common human pathogens that contributes significantly to the elevated fatality rate associated with infections. Infections in humans resulting from bacterium Staphylococcus aureus is vary from minor skin injury to more severe conditions, such as bloodstream infections or pneumonia. The significant pathogenicity and multi-drug resistance of these bacteria are its origins. Regrettably, Liu et al. 2017 indicated that the common genetic modification tools used to look over the suitable molecular mechanism of Staphylococcus aureus have several limitations, such as long experimental period, relatively low gene editing efficiency, and leaving scars in the genome. Therefore, the CRISPR-Cas9 system has been widely utilized across a range of organisms, including bacteria, as a tool of genome editing tools. This is because the CRISPR-Cas9 system offers rapid and effective manipulation of chromosomes in Staphylococcus aureus. Furthermore, as outlined by Liu et al. 2017, the deletion of TGT and ROCK-A genes and the introduction of the ERM-R cassette into the Staphylococcus aureus are the example of the functionality of CRISPR-Cas9 vector. Finally, this research established the CRISPR-Cas9 genome editing tools in Staphylococcus aureus, enabling fast, scarless, and marker-free genetic manipulation. I will explain one of the procedure, which is hemolysis assay. First, grow the Staphylococcus aureus culture in TSB at 30 degrees Celsius overnight. Next, the culture of the strain was loaded onto the agar plate. After 24 hours of incubation at 30 degrees Celsius, the agar plate was observed. Okay, next, the real-time PCR was performed. 5 microliter culture of Staphylococcus aureus strain was incubated at 30 degrees Celsius. When the optical density reached 1.0, the cell was harvested by centrifugation and lysed at room temperature for 40 seconds. The RNA was isolated by using RNA ISO+. The RNA was reverse transcribed to cDNA by using PrimeScript-RT reagent kit. To complete this step, we need to do the quantification of cDNA by using the Lightcycler 96 system with SYBR Premix X-Tech2 kit.
Fascinating! Now, without further ado, we're gonna welcome our young biology researcher, which is Dr. Ain Yohanis, to explain more about the procedures on CRISPR-Cas9 that was prepared by her.
Thank you to the MC. My name is Ayuhanis and I will continue to explain about another procedure on performing CRISPR-Cas9 mediated gene editing in Staphylococcus aureus. Today, we will talk through the key step in designing the CRISPR construction, preparing the bacteria strain and performing the electroporation. For your information, in the bacteria, the cRNA and tracer RNA are separate molecule entities. The discovery showed that these two molecules can be combined into a single molecule by fusing them together with a linker to generate something called single guide RNA or sgRNA. If the sgRNA complex with a Cas9 protein, these two component system will be able to cleave DNA just as the three component system does in bacteria. So, it is possible to determine any sequence of 20 base pair as a target for editing. And for that to be done, is just need to synthesize the appropriate sgRNA with the complement sequence and then insert that into a cell along with Cas9 protein, which has been short formed as aureus. Now, let's design the CRISPR-Cas9 construct. This involves selecting a target sequence, design a single guide RNA sgRNA, and clone it into a suitable plasmid. Next, we prepare the aureus strain for transformation. This includes growing the bacteria and make them competent for electroporation. After that, we will perform the electroporation to introduce the CRISPR-Cas9 plasmid into aureus by mix 1 or 2 microgram of CRISPR-Cas9 plasmid with 50 microliter of competent aureus cell and then transfer the mixture to a pre-chilled electroporation curve with a 2 mm gap. After that, set the electroporator to 2.5 kV, 100 ohm and 25 microF and pass the cells 1. For the recovery, immediately add 1 ml of SOC medium to the curve and transfer to a sterile tube and incubate at 37 Celsius for 1 and 2 hours with shaking. Lastly, add 100 microliter of recovery culture TSV agar plate, contain the appropriate antibiotic to select for transformate and incubate overnight at 37 Celsius. Finally, we confirm the successful gene editing in aureus by picking several colonies and perform colony PCR to screen for the presence of CRISPR-Cas9 plasmid and the desired gene. And that's how we complete the CRISPR-Cas9 mediated gene editing in aureus. This powerful technique allows for previous genetic modify and open new avenue for research and biotechnology application.
Absolutely! It is a complex landscape. CRISPR-Cas9 holds immense promise for treating genetic diseases, enhancing agricultural practices, and even contributing to conservation efforts. However, ethical concerns about gene editing in humans and ecological implications in altering organisms should be carefully navigated. Well, viewers, buckle up as we embark on this enlightening journey into the world of CRISPR-Cas9. Stay tuned for an in-depth of explorations and surprising insights. You're watching BioAds and stay tuned for the next show.